Good evening, everyone. Welcome to the Network Seminar Series hosted by the Center for Networked Intelligence at the ECE and RBCCPS IISc. It's my pleasure to introduce today's speaker, Professor Hari, and he will be speaking on aging brain research, challenges and opportunities. Professor KVS Hari is a professor in the Department of ECE, Indian Institute of Science, Bangalore. He holds a BE ECE degree from Osmania University, Hyderabad, MTech in Radar and Communication Systems from IIT Delhi, and PhD in System Science from UCSD, and has been a visiting faculty at the Stanford University and affiliate professor at KTH Royal Institute of Technology, Stockholm. His research interests include 5G wireless communications, radar systems, autonomous vehicles, neuroscience, and affordable MRI systems. He is a co-author of IEEE 802.16 standard on wireless channel models. He is a fellow of INSA and a fellow of IEEE. He is currently the director of Center for Brain Research at IISc. Over to you, folks.
Thank you. Good afternoon. So this talk will have no equations. Okay. It will be more about an overview and what engineers can do for healthcare, medicine. Okay, so that's how it is. So the title is, of course, Aging Brain Research, Challenges and Opportunities. Taken wherever the source has not been is not mentioned in this slide, it's from the internet, and as the book Principles of Neural Science, which is sort of like a several hundred pages thick book, which I used to study when I took the course on Introduction to Neuroscience several years ago, almost a decade ago. Okay, so the first term in that title was aging. So we need to understand what is aging, right? And we all start with cells, right? So healthy and aging cells, so what exactly it means? So what is a healthy cell? Basically, a healthy cell has the ability to divide, produce energy, involve in waste removal, nutritional uptake, and so on and so forth. So that is a healthy cell which is able to divide. This is very important for you. There is a limit called the Hayflick limit, which refers to the maximum number of times a cell can divide, somatic cell, before it permanently stops and enters a state of senescence. Okay. Now 40 times, so the human being is, the cell in a human being can divide 40 to 60 times. Different cells have a slightly different clock. So therefore an average living human being can sort of be about 80 years. If cells don't, cells are able to divide further, then you live longer. Okay. Compared to the turtle, compared to the humans, the Galapagos turtle can divide up to 110 times. Okay, and therefore it can live much longer. So when you say when each cell can divide 40 times, it is what? 2 to the power 40 kind of cells you can generate, right? So that is what it is. Now the aging cells, when they stop dividing, but they are metabolically active. Okay. So what does senescence mean? So cell senescence has been defined as an induced irreversible state. So once it is, gets into this stage, it is irreversible, and there is a reference for that. They don't die, but actually they secrete substances that affect healthy cells, right? So that is bad news. So not only are you aged, you create trouble for the younger guys. Something similar happens in many institutions also. So recently, at Stanford, researchers found some consistent nonlinear patterns in molecular markers of aging, where they found that at two distinct points in chronological age, around 44 years and around 60 years, there is a sudden change in this accelerated aging. Okay. This is based on a small cohort, about 108 people. They have done from 25 to 75 and looked at different things. They did omics and other things, and they found this. So it's a bit of a interesting outcome that at the age of around 44 years, 45 you can take, and age of 60, there is a change in the way the cells start to behave, the healthy and the aging part. So all professors need to be worried about it, need to be taken care of. 45 years typically is what it happens. So this is a paper you can look at. Okay. Now this is, so what happens when a cell divides? The genetic content of the cell gets replicated, correct? So this is an image of the chromosomes. Now if you notice, the blue colored things are the chromosomes, and at the end there is a protective cap, as they call, the green ones, which are called telomeres. Okay? These are very important things. So this is what is, so what exactly those telomeres are doing is to avoid these chromosomes to get connected to each other or to become sticky, etc. So they are protective caps. Now what happens when aging happens, right? So aging or division, healthy cells, forget about aging, healthy cells, when they divide, in the replication process, not everything of the chromosome is replicated. A part of the telomere is not replicated. Okay? So every division reduces the length of the telomere. Okay? So in some sense, the length of the telomere is sort of thought as a reasonable marker for determining the age of your organism. Okay? So this is very important, because as when the telomere is unable to, is not there, let's say shorten, and the shortening can happen due to various reasons. Shortening can also be prevented by an enzyme called telomerase, but if that is not there, in some cells there is a, in cancer cells it happens differently, but in general, so the cell will die or it will become senescence. So when it becomes senescent cell, it can no longer replicate, but it will create trouble because it secretes something called SASP. Now in our body there are many cells which will divide, but there are some cells which don't divide. Neurons are one such type of cells. They don't divide. Okay? So it's interesting. So therefore now we can feel that, okay, neurons are not dividing, so therefore there is no problem of this telomere business, and therefore there should not be any aging. Correct? If they don't divide, there is no problem, right? Yeah. We'll see what exactly happens. And the other are RBCs and cardiac. Okay. So aging is over, now brain. Okay? So this is just a functional representation of the brain, where you have different parts of the brain at sort of the allocation of different tasks and functions of the human brain. So there are different parts. You have the visual part at the back of your head, the attention, planning, decision making is in the front of that, of your head, then there's movement, touch, taste, hearing, speech, smell, and so on and so forth. There are other areas. Memory is there, hippocampus. Okay. So for some of you who may not know what the brain has, there are different kinds of cells in the brain. So we all are familiar with the neuron, correct? Neuron has a body, then there is an axon, and then there is a myelin sheath, which is important for the communication. I will explain to you. Then there are other cells called glial cells. But it does not do anything like that, but it has that. So, glial cells have different, so there is different kinds, okay, astrocytes, microglia, oligodendrocyte, etc. So, what do they do? Astrocytes are basically work towards nutritional support for the neurons, blood-brain barrier maintenance and ion balancing. Okay. Oligodendrocytes are axonal insulation, so they take care of this sheath, and I will tell you why it is important. And microglia does debris cleanup. Okay. So, all these chemical reactions which happen, there is a debris and that has to be cleaned up, right, and that is what this microglia does. So, when you are born, 99% of the neurons are formed before birth, because neurons do not divide, correct. But in the process of when the fetus is there, most of it, there are a few regions in the brain which allows some neurogenesis, especially in the hippocampus area, etc. So, most of the neurons are in the brain when you are born, and they do not divide. Mature neurons do not divide. So, what happens when, why is it that we have a different size of the brain when we grow, right? So, all that is happening because of the other cells which can divide. So, the glial cells will divide, and that sort of creates the size of the brain. Okay. So, just an example of some different kinds of neurons, so we do not worry about that. So, the retina is considered as an extension of the brain, okay, because there is an optic nerve and it is considered to be that. So, you have let us say this is the rod cells and the cone cells, so you must have heard about it. Then you have different kinds of neurons and it touches the optic nerve. Okay. These are just two examples I am giving. So, what happens is when, let us say you touch something, okay, the signal will go to the spinal cord first, and then it goes to different parts of the brain. This is just an illustration of different parts of the brain, the medulla, pons, midbrain, and this is the region of the brain which is handling your touch. Okay. Similarly, hearing, if you hear, then it goes into different parts, and this is the part of the brain which is for hearing. And there are different vision, etc. This is just as an example. So, there are pathways in the brain when you do some function, okay, which are useful for cognition. Okay. So, since this is an ECE seminar, there should be some resistor value ECE electrical network. So, people have modeled this. So, what we have in the brain, in the neurons are what are called ion channels. Okay. So, this side is the inside the neuron and this is the outside the neuron. Okay. And there are ion channels which are sodium, potassium and chloride channels. So, these have been modeled where you have a source, a voltage source of a certain value, and then there is basically G is conductance is what they have. So, units of conductance is basically inverse of the resistance. So, the new unit, or if you are not aware, is Siemens now. Okay. Previously we used to call it mho, MHO, reverse of OHM. Now Siemens is the standard SI unit. Okay. So, this ion channels can be modeled as an electric circuit. Okay. So, that is just one. Now, let us see how the signal travels. Okay. What exactly happens when you have a neuron firing, we call it, right. So, just imagine that this neuron has lot of dendrites, these are called. They will be connected in some sense to other neurons, right. So, there are certain neurons which are excitatory neurons and certain neurons which are inhibitory neurons. So, what happens is there is something called a resting potential based on the ion channel concentration, sodium and potassium. Okay. So, this resting potential of a neuron is at -70 millivolts. Okay. So it goes on. Then when some other, the neighboring neurons start to communicate with this neuron, they could be excitatory or inhibitory. The sum total, it's a superposition principle, will be the trigger here, okay, at some point in time, whenever that thing happens. So immediately the ion channels get activated. So the sodium channels will open up, the potassium channels will open up, close, and there is a sudden burst in the action potential, as they call. So sodium ion channels will start to flow in. And then there is a repolarization, there will be a potassium channel which will be going out. So there is a peak, the action potential can go from -70 to +40 millivolts. So it's a big spike, right? And then there will be a hyperpolarization, goes down and then comes back to resting state. Now this peak has to now propagate. So how does it propagate, right? So this is a peak which has been established. So this propagates across this by a sequence of ion channel modulation. So the ion channels which are there along this path of the nerve, the axon, are opening, closing, so the wave propagates. So the spike propagates. And the sheath is important, the myelin sheath, because the myelin sheath does not allow ion channel. When there is no sheath, it has to activate ion channels. So it takes time for that. So it slows down. So but the myelin sheath is not allowing those ion channels there, so it just propagates. So it speeds up the transmission of that pulse, whatever action potential. Now, what happens at the end? So I would call it, it's a wired communication signal, right? It's a wired. So the signal is traveling through the body of the cell. Then when it comes to the end, there is no connection to the other neuron, to the neighboring neuron. So there is a gap, so wireless, right? So there what happens is calcium channels get activated. This potential will open some calcium channels. It will trigger some neurotransmitters. Those neurotransmitters are sent and they are received by the receptor neuron, by receptors on the neuron, and then there is again a potential change because the ion concentration is changing. Okay, everything is that. So it is a electrochemical process which allows that signal to traverse different ones. So it arrives at the axon terminal, voltage gated channels open, calcium channel enters the presynaptic part, and then they go on, and then the signal is initiated in that. So once that happens, it will again trigger in this neuron this part, and then there will be again an ionic exchange, and the peak is there for it to transmit. Okay, this is the electrical network in the brain which is there. So now, if the neuron is not dying, and if this communication is happening, what can cause loss of communication, right, is the question we can ask. So if suppose due to some reason this sheath, this myelin sheath gets demyelinated, okay, because the glial cells were supposed to clean up the debris and they are not functioning, and the chemicals are trying to rip the myelin sheath, okay, then what happens? The signal will take much longer to traverse, correct, because now that speed has been reduced. So it will take some longer time, and the ion channels, etc., may be moving, not opening and closing will happen. So that is one possibility of lack of the connection. The second one is there is a gap here, correct. What if something obstructs the transmission of the neurotransmitters going into the other synapse, suppose, right? That can also cause a break in communication. Okay? So, these are the structural issues which one has to look at. The other one is, what if there is something here which does not allow this thing to go? Assume ion channels are still working, but something is stopping this electric wave because there is some foreign thing which is inside which is stopping this wave to propagate. That is also one possibility of the circuit or the network not working. So, keep this in mind, and then let us see what we do. Now, let us define what is cognition, right. So, cognition is, according to the definition of the American Psychological Association's dictionary, all forms of knowing and awareness such as perceiving, conceiving, remembering, reasoning, judging, imagining and problem solving. Okay, it has almost everything there. Now, what happens when there is a decline? Why are we not able to do due to healthy aging? Let us say we have no disease in the brain, but healthily we are aging. Why are we healthily aging? Why are we aging at all? Because some of the cells which became senescent and they have stopped dividing, and if you stop dividing, there are no fresh cells. If there is something, damaged cells are there, so you cannot repair. Okay, so there is a decline. Now, cognitive decline due to healthy aging is neurons do not divide. So, the healthy aging due to, the cognitive decline seen in healthy aging is not caused by neurons at all. It is not caused by the functioning of the neurons. The neurons are intact, but it is the support system, the aging glial environment, which is not doing the providing nutrients, insulation, cleaning services, all that is there. So, the neuron is there, and if it is not nourished by the glial cells, then the neuron will die. Okay. So, the neuron is not causing any trouble. It is a very intact structure, but the support structure is creating this trouble. Okay. So, the glial cells are, just to give you just a recap, that is what I did. Okay. Yeah.
So why wouldn't senescence happen if not for cell division, if they know the cause for senescence?
So, cause for senescence is, as I told you, the telomere length, right. So, that is one of the causes. Then there could be other causes where because there is all a chemical, a pot of chemical reactions happening. So, sometimes the cells will die due to.
Why can't that happen to the neuron?
That is the structure nature has created. Neurons will not divide. So,
No, my point is,
yeah,
neurons won't divide, so that's one cause electric.
correct.
So there could be other causes also, right? Not just that.
There are no other causes. So far, the science has not found other causes. So, yeah. So there are some other maybe minor thing. So, the neurons, I will tell you what happens. There are other causes which can cause problems. So, because things are not happening in a healthy manner. Yeah. Yeah. Yeah, brain function is different. The glial cells are not restructuring, they are there, they are dividing, and they are trying to address the requirement of the neural networks.
Reconnecting, I mean.
Ah.
They are reconnecting.
They don't reconnect. The neurons are the ones which reconnect.
Neurons are the ones.
Yeah.
So that may be the aging can be part of that also, whether they are able to reconnect or not.
Correct.
His question was on.
Yeah. The ability to reconnect is facilitated by the support network, because it has to maintain the ion concentration. Only then it will enable these things to happen. So that's what it is. Yeah. Okay. So this is a cartoon picture of what is cognitive. How does it help? Right. Suppose you plot the cognitive decline on the y-axis and time in the x-axis, and this is, let us say, healthy aging. Okay. Just a inclined line. It is not like that, but cartoon. In a disease, when there is a disease of the brain, so it will follow up to a certain time age, the brain aging as a healthy decline, and then slowly it will start to follow like a waterfall curve. Okay. So, the disease is altering the slope of this line, and this slope of the line can be different for different people. Okay. It could be due to genetics, it could be lifestyle, it could be maybe comorbidities, you have other diseases, so it could be that. So, the key is this part called mild cognitive impairment, MCI. Okay. So, if I am able to detect this early, I mean I have to detect this, right, then I have a chance to not slip down this slope. Okay. So, this one, our job is to see whether if I detect this, then I can pull this curve up somehow by intervention, what is called intervention. Okay. No, it is
MCI.
here.
Oh,
Yeah.
okay.
This is disease.
Okay.
Okay. So, this is here. So, when there is a disease in the brain, then it sort of accelerates the aging process sooner.
Oh, but in the first two slides you mentioned like at 44 years and 60
There
years
is
there is
a
change
time
in
point where aging is getting accelerated.
Okay, but
That's
but
all. So, which is
here-
causing that green line if it is healthy, in healthy
Okay,
people.
okay.
Yeah. Okay. And in India, this burden is going to go. We are all going to become old, and right now we have a demographic dividend where youth is, the average age is quite low, but in another 20 years it will be an aging population we should worry about. So, dementia. Okay. So, this is a WHO report, which is a blueprint for dementia research. So, dementia, normally people think that it is only loss of memory, but actually dementia is an umbrella term for a set of diseases. Okay. So, Alzheimer's disease comprises 60 to 80% of dementia. Then there is vascular dementia, dementia with Lewy bodies, frontotemporal dementia, dementia due to Parkinson's disease, So, Parkinson's disease can accelerate dementia. And all of them are basically due to some protein pathology, which I will explain using a figure. Okay. So, there are plaques and tangles, etc. This vascular dementia is cerebrovascular pathology. So, in India there is a lot more vascular pathology, vascular dementia occurrence is what people are observing. So, what is dementia? Well, day-to-day forgetfulness, problem with language, difficulty solving problems, etc. If it is happening on a consistent basis, then it is dementia. We all forget things, we all misplace things, that is not dementia. Okay. So, you should not be worried about that part. Occasionally if you do, yeah. So, dementia is like let us say you have, okay. So, what happens in memory? The hippocampus is the place where the short term memory is stored. Then the long term memory is stored in the prefrontal cortex. So, sometimes you remember old what has happened 10 years ago, 15 years ago, if the disease is not affecting that part of the brain. But if the hippocampus is getting affected, then you start to forget even short term things. Okay. So, that is what it is. So, what exactly happens in dementia? What is the pathology of disease in Alzheimer's, let us say. So, let us assume that, let us say that this is the healthy brain, okay, where the neurons and the dendrite, axon, the dendrites are all there. In the Alzheimer's disease brain, which is basically you have what are called plaques. Okay. So, and because of that plaque, the communication is not happening, the nourishment to neurons is not happening, therefore, the neurons start dying and there is atrophy in the brain. Okay. These are based on slices of brains of people who passed away with the disease, people who were able to find out what is exactly has happened. Okay. So, if you go through this carefully or in more detail, so now you think about this neuron, something you see here called tau. Okay. These are tau tangles which is inside the neuron. These are outside the neuron amyloid beta plaques, which are basically stopping the neural synaptic communication. That is what we saw, right. If something happens in the gap, you can stop the communication. If it happens inside, maybe the signal is not traveling. Now, what are these two they are misfolded proteins which stick together. So, a protein is a three-dimensional structure. Okay. Now, if the folding operation has an error, then they do not lock with each other. Okay. Proteins are three-dimensional, they lock with their counterpart. Okay. But if they are misfolded, then they are not able to lock, they start sticking to each other. When they start sticking to each other, they form plaques or tangles, they are called. So, here they are called tau tangles, amyloid plaque. Now, these things take a lot of time for this to keep sticking, stopping the neural communication, and they start moving across the regions of the brain. Okay. So, the issue is that when this happens, there is a cerebrospinal fluid in our brain, okay, which is where all these other cells are there. So, these tangles are outside, these plaques are outside. So, the cerebrospinal fluid can be used to detect the concentration of this. So, the technology which has been used is PET, amyloid PET. So, what do? They have an item, a radioactive tracer compound which binds with the amyloid beta plaque. When it is injected, you will get an idea of how much of amyloid beta plaque is there. Okay. So, that tells you and that is the final gold standard clinical decision which a neurologist will make if the patient has Alzheimer's or not. That is the final decision, because we are actually seeing the concentration of plaques in the brain. Now, similarly, if you are able to get the tau tangles concentration, right, because what will happen is this is not sitting there. Okay. It is going to start moving and infect the other one, the next synapse. And it has been shown that amyloid beta can accelerate this tau tangle propagation also. So, the time it takes for these tangles to form and affect your neural communication pathways is 15 to 20 years before you are diagnosed clinically. So, when somebody is diagnosed with dementia today, the disease started 15 to 20 years before, but there were no symptoms. Okay? And that is where we need to find a way to understand how quickly can we estimate this mild cognitive impairment much before the clinical diagnosis happens. Okay? Once you do clinical diagnosis, it is too late. There are no drugs as of today which is a cure for Alzheimer's. There is no cure for Alzheimer's, and that is the challenge. It's a global challenge. Okay? And since it is a very slow moving disease, even research is difficult, because if you want to study the effect of something, it takes time to do that. Okay? Okay. Now, these cognitive decline, all these protein biomarkers, I mentioned in the previous slide that while CSF has these, blood plasma, yeah. Similar. Yeah, that's why we use mouse models to understand the effect of drugs and the pathways, trying to understand even how amyloid plaques are formed, etc.
So, the 15 years that you mentioned is for humans.
Human.
For the rats,
it is much few months.
Few months maybe.
So, they
so
have transgenic mice. We have transgenic mice in our animal facility in IASC, where they exhibit memory loss. So, they are born. After some time, the AD plaques get propagated. After a few months, they are memory loss. So, what is the experiment we do on mice is the following. So, this is called a freezing time experiment. So, you take the mouse and put it in some environment. They are breeding in one environment, but you take it to an experimental environment. Then when it is there, you trigger some shock for 1 second, 3 shocks you give. Okay? It is moving around and then you give shocks. Then you bring it back to their home. 24 hours later, you bring that mouse back and present it to the same scenario. So an AD mouse will not remember it was shocked, so it will keep moving. So we measure the freezing time. But a normal mouse, which is called wild type mouse, knows that it had shocks last time it came. So it will stop moving, or it will move less. That is called freezing time. So this is a way we people are trying to understand. And then what you do, if you have these AD mice, you want to try, let's say, a drug molecule to see whether it can do something. You inject that drug into the mouse and then see whether the freezing time it improves. If so, then you sacrifice the mouse, take the brain of the mouse, slice it, and start looking at the cellular level histology of them.
So there is a hierarchy of this kind of a aging period for higher mammals and other things, maybe monkey or.
Monkeys don't have Alzheimer's apparently. Some of them don't get this disease, so that's the challenge for us.
Or even elephants.
Yeah, elephant, I think. I don't know that part.
Nature is.
As a biologist, I can excuse myself as an engineer of ignorance. Yeah. Yeah. Misfolded.
The dirt that usually is being cleared by the
Yeah, so the
sheets.
glial cells cannot clear it. That's what your question is?
Yes, yes.
Yeah, they cannot clear the plaques. Yeah,
Okay.
yeah, correct. They have no mechanism of clearing them.
Is it because that is huge in size?
They cannot bind to it, or they cannot do anything to clear it up. Yes, correct. Yeah. Yeah, correct. Yeah. So plaque are basically sticky items which are causing disruption in the communication. Okay. Okay. So so now the question is, if I wanted to know 20 years ago how much of amyloid beta plaque is there in the brain, you had to do PET imaging. The quantity of that protein in cerebrospinal fluid is high, but because blood is also traveling there, these proteins are seen in the blood. But 15-20 years ago, the concentration of these proteins in blood plasma was very, very small, and there were no techniques. So it was a signal to noise ratio problem. Okay, low signal to noise ratio to detect that small quantities. Now technologies have emerged where they are able to detect, the concentrations are a few fractions of picograms per milliliter. That is the level in the blood plasma. CSF will have higher concentration because it is there right at source. So now the question is, now the technology has developed where they are able to amplify the strength of that signal, and using fluorescence techniques they are able to detect the quantity of this with great accuracy and in a multiplexed manner. Okay. So in the blood plasma, there are many proteins, okay, and these proteins can be caused due to oxidative stress, platelet dysfunction, axonal degeneration, astrocyte. All these are proteins which are there in the cerebrospinal fluid and which are markers for degeneration. Okay. So it is good for us to understand, the challenge is to understand what is normal healthy protein concentrations and what is for MCI and what is for disease. It's a classification, right? I mean, it's like a hypothesis testing problem. You have to detect, suppose somebody gives you a protein value of this or several proteins, a few hundreds of proteins, how can you form a good function to estimate what is the cognitive decline? That's the problem which we have to face. Yeah. From the spinal cord called lumbar puncture and bottom. The problem is that for patients they are willing to take, For healthy people, they are not willing to give, because it can cause, you are actually piercing the spinal cord. Any problem, you are going to induce some other problem. So, diseased people are okay to give because they are already diseased, they want something to happen, but healthy people, very difficult to get lumbar puncture. So, the vertebrae, the spinal cord is in between that. Yeah. So, there are gaps in the vertebrae. So, you have to, and if you do not know, if you are not doing a good job, then you are in trouble. Okay. So, now all this is fine, all that. Now, Lancet study has shown that for dementia, there is a certain, these are called modifiable risk factors. Okay. So, early life, less education, suppose you educate people with education and no education, no formal education, okay. People are educated even without going to school, but in this case, if you are going through so many years of schooling, then if you can educate people in their childhood, you can reduce the number of cases of dementia by 5%. That is what is the global study. This is a global study. Similarly, in midlife, hearing loss. Okay? If you can address hearing loss by designing good hearing aids which are comfortable for somebody, you can reduce it by 7%. Cholesterol, if you can control, 7%. Depression, 3%. Traumatic brain injury, like motorcyclists and scooterists who don't wear helmets and get into accidents, traumatic brain injury, you can reduce. Physical inactivity, if you are not doing physical exercise, you are very sedentary, not good. Diabetes, 2%. Smoking, hypertension, obesity, excessive alcohol. So, all these are modifiable risk factors is what Lancet has said, doing a cohort study. Late life, social isolation is very, very critical. So, I will show one slide later if I have time. Air pollution, visual loss, etc. Okay? So, 45% of dementia cases can be reduced if we address these things. Okay? So, it is good for, so this is a motivation for us to see, okay, what is the motivation for us to do any studies? How do we address this problem now, right, in a population way? So, the WHO report says that we need high quality data, understanding the underlying diseases using biology and chemistry and neurochemistry, neurobiology. We need to build biomarkers, right, so that I can detect that. And then of course, you need to understand risk factors. I have just highlighted a few, but there are so many other things, right. So, this is what it is. So, what do we do then? What will be a good set of biomarkers, correct? I think I had a slide which, did I define cognition before? Ah, yeah, good. Yeah. Okay. So, biomarkers, basically what we need to know is what is the lifestyle, what is the demographic information, education, all that we collect. Then a doctor has to understand basic history, medical history, etc. We collect that data. And cognitive assessment. So, there should be a tool which is basically doing the cognition, right. How do you measure cognition? How do you say that this person is cognitively healthy or not? There are cognitive tests which are standardized. Then you collect blood, then you do biomarker, then you do genetic analysis, because there has been a connection with genes and genome and the disease for other diseases. Cardiovascular parameters, eye, because we said, right, retina is an extension of the brain. So, can I see something in the eye, in the retina, which can tell me what is the neurodegeneration happening in the brain, right? These are markers. And then of course, cardiovascular pulmonary function, MRI, you would like to know how the brain structurally and functionally. Gait, because that is neurodegeneration, if you are not able to walk or there are changes in your gait. And then hearing and so on and so forth, right. So, these are markers. Some of them are fluid blood based and some are digital. we'll call them digital, right. So, conventional markers, and then there is a digital assessment battery, and you can do proteomics on that. All these and many more need to yield or lead to a composite multimodal biomarker. So, it is a sensor fusion problem, okay, for engineers, right. You have an estimation of a variable called MCI, okay. There is something which gives you a quantification of cognitive impairment. It is called clinical dementia rating score. If CDR is 0.5, that means you are MCI. Now, you are given this task. You have on the left side all the variables from each of these domains, okay. Let us say you take the retinal fundoscopy or do the optical computer tomography. It will give you, let us say, 100 parameters. It will tell you the thickness of your retinal or the optic nerve and the various parameters. So, that is one. Using this 100, and if I tell you by this cognitive test which I have done as a gold standard, that there is a MCI for, I have data for normal MCI people, right, and disease people. And if I do this, it is a problem of trying to find what is the best model that can predict or estimate the MCI, given I have information about the ground truth, correct. So, you can use regular statistical models, or if you are all into AI fever, you use AI models, okay. So, each of these can give you a time series. You do it longitudinally, because today you may be normal, after 2 years you may become MCI. So, I need to estimate in the using the time series whether I can predict the MCI for some people, right. Then it becomes a good functional thing. So, while this is all looking like some biology part, but actually there is a lot of parameter estimation, statistical estimation, hypothesis testing problems. These are all that, okay. So, at CBR, yeah. So, clinical dementia rating is a score which is an assessment which the clinician does when they meet that. Yeah. Not, no, that is the opportunity for us to actually do lot more, because other cohorts people have done something, but not. It is about 70–80% prediction models are there now, but we need higher. Yeah, confidence. But it depends on how much data you have, and it is population based. So, the uniqueness is for us is to understand the Indian population, because we do not know what the model is developed on Caucasians. For example, genetically there is something called ApoE4, which is linked to dementia in the Caucasians. Then recently they have studied in African Americans, this ApoE4 is not connected to dementia. Okay? It is not giving the same results of connection to that. Now, in India, we have lot of African descent. So, we are in India, if you look at the genetic diversity, India has 4,600 ethnic groups. Recently, DBT a few years ago funded a project called Genome India, where they sequenced 10,000 human genomes. That means 100 ethnic groups, 100 each were sampled across the country, including tribal regions, etc. And a broad categorization is that you have a European ancestry, you have the African ancestry, you have an Australopithecus, that is our ancestors, that ancestry, and Tibeto-Burman ancestry, broadly. Dravidians, all that are there, they are all mixed. There are 130 million new variants found in the Indian genome, which are not there in Caucasians. So, it is very important that we collect data of India to understand what is happening. What will be these protein concentrations? We do not know, right? Proteins are basically what you eat, how you exercise, what are the comorbidities. So, it is very important that we collect data which is required for us to build these models for India. right, Indian population, and we are 1/6 of the world's population, so we are impacting global health in some sense. So we have two cohorts. The urban cohort funded by Tata Trust called Tata Longitudinal Study of Aging started in 2015, 1,000 subjects we follow every year. So all those biomarkers which I made a list, every year every subject walks into CBR and we collect this data. I was part of this cohort till I joined CBR. Because of conflict of interest, I was removed from the cohort. So for 5-6 years I actually gave this data. The data is there, but they are not using it. Okay? So we do that, follow up. So this is a high literacy cohort, more multilinguals, more genetic heterogeneity, because people from all over the country are here. Spurred by this, in 2018, CBR started a cohort rural where the target was 10,000 subjects. We wanted to track 10,000 every 2 years. Okay? That is an agricultural community in Srinivaspura in Kolar district. 100 kilometers from here, Srinivaspura is a taluka which has about 200 villages, and we collect data by going into the villages. We have been collecting, and 2025 we met the target of 10,000 first visit. We have 5,000 second visit. Okay? So we have this longitudinal time series now. Now in our area of work, you generate thousands of time series by using a model to simulate, right? But here is a social system. It takes time to do that. It's very difficult. And less migration, more mono and bilinguals, and less genetic heterogeneity because it's an endogamous group, right? People are marry within each other, follow up. So that is called SANSCOG, okay? And above the age of 45. Anybody above the age of 45, we have a 100-year-old also in this cohort. Okay. So this is the infrastructure we have where we collect data. So it's a completely sensor based, all engineers should be. So we do have this OCT, optical computed tomography, we do audiometry, we do tympanometry, we do audiometry, the frequency response of your ear, gait assessment. There is a gait mat full of sensors, you walk on it and then you give a cognitive load. For example, you walk normally and then they'll give you a task. Spell the word black backwards while you are walking the next time. Okay? Your stride length, your velocity of your walking, everything will change because you are now have a cognitive load. Okay? So similarly, we have autonomic function testing, spirometry, we have carotid Doppler. Carotid artery is basically trying to get the blood to the brain. Okay? EEG. Then we have genome sequencing machine. We have a biobank which samples, sorry, which stores all plasma samples at -80 degrees centigrade freezers. They can last. You can do research even after 40 years. Okay? The plasma will be there. So suppose tomorrow you get a new protein biomarker, I can go back to my biobank, pull it out, and I know the sequence of the cognitive assessments of all these subjects. Let's say 5 years, they were healthy, then they suddenly became MCI and maybe diseased. I can then analyze all that and get the data to do something. 3 Tesla MRI is behind us, and we have a storage and high-performance computing cluster. We have about 5 petabytes of data total, including genome, etc. So recently we started a biomarker center of excellence which does proteomics. So this is a graph which tells you at the time of symptom diagnosis, these kinds of proteins which are called phosphorylated tau, etc., start almost 25, 20, 15 years before. Okay? So the goal for us to see whether we can detect these concentrations, first we need to know whether these concentrations belong to normal category, MCI or disease, for which you need to collect data, do a normalization, collect and see for Indian population what is the value. For diabetes, we have HbA1c is a glycosylated hemoglobin. You know that if it is less than 6, you are not diabetic. It is 6 to 6.5, you are pre-diabetic. After that, it is you are diabetic. They will declare. Something like that for brain health, we need to build. It is not yet there. FDA has approved p-tau 217 as a diagnostic biomarker. last year. Okay. So, we need to see what are the values for India, correct. We have to collect data. So, this is a machine called Alamar, which is a new technology which can detect these few picograms per milliliter concentration of proteins. So, it detects 131 proteins related to neurodegeneration and 250 proteins related to inflammation. Inflammation can cause neurodegeneration also. So, we need to understand those proteins. And this is the first installation in India. We have 2 machines actually funded by Gait Ventures. Okay. So, quickly about our cohorts, we have, as I said, the age group of the rural is red and urban is blue. So, you can see age group, you have people from 45 onwards till, as I told you, even 100-year-old. The recruitments, so we had recruitments which had a dip during COVID, we started small and then we have achieved that. So, if you, this is a good thing for TLSA urban, right, started in 2015. Every year we are adding new people. V1 means all new people, first time visitors. So, we, even though 1000 was our target, we have done up to, we currently have 1670 because many of them drop out. Okay. So, there is attrition. Then V2, so we have 1000 people data, this is up to 2025, and V7, V8 we have 14 people, V9 we have 6 people, like that. So, for the last 9 years they have been coming. Okay. So, for SANSCOG we do every 2 years, so we have less number of visits because we started in 2018, so we have very few visits, but we still have a good 5000 people in the second visit. I think occupation, yeah, mostly agriculture, rural and technical and professional in the urban. Okay. Education levels, illiterate, 50% of the rural population is illiterate, and then we have of course higher degrees there. This is the clinical dementia rating score, as I said. So, 0 means normal and 0.5 is MCI. Okay. What has been found is, in urban population, whatever 1000 people, about 8 to 9% of them have MCI, developed MCI. So, a few hundred, that is all, around 100. But in rural it is 18%, double almost. So, we have more because 10,000 people, 18% MCI. Yeah. So, each visit generates about 5 GB of data. If you don't do, if you do a whole genome sequencing, which you do only once because that does not change, 70 GB. So, we have about 2 petabytes of cohort data and we have another 2-3 petabytes of the genome data of the Genome India project. So, this is what it is. So, what is the way forward for us? Right now these are healthy cohorts. So, we have these cohorts here in this part of the, some of them we have MCI and maybe some of them healthy people, right. We have 70 years, 80 years, healthy people. But what is missing is here. Okay. We need people here, only then I can classify healthy, MCI and disease with all this, right. So, the goal is that we are going to build a national network for brain aging. So, we are going to build a national platform. We are going to connect with other community cohorts, hospitals, other cohort, clinical wellness centers, everything. And we need diversity also. We can't just define our biomarker based on Bangalore and Srinivasapura Taluka, right. We need diversity. So, we have already signed MOUs with Madras Diabetes Research Foundation. So, there are many cohorts in India, but their objectives are different. They want to understand diabetes, they want to understand cardiovascular. There is a cohort funded by NIH in India called LASI, Longitudinal Aging Study of India, of which they sampled about 10 years ago 75,000 people, they interviewed and they got their blood sample and stored. There is a dementia subgroup of 4,000 people which they have done cognitive tests and collected their blood again, right. So, that is the information, very valuable. We are going to do the proteomics for LASI-DAT, about 5,000 samples of proteomics we will do. And the proteomics, as I said, there are 131 and 250, so 380 proteins for every subject, every visit you will get. So, huge data we need to do. So, what we want is robust risk scores and dynamic trajectory trackers, and we will be doing that. So, the goal is this. Now, the question is, do we need to wait for 1 year to get data for seeing the change, correct? It's going to be a low frequency sampling, correct? So now with variables, and we know that every sensory function what we have can be now monitored, let's say, right? So we are embarking on a multimodal digital platform where we will look at speech, we will look at sleep, we will look at the eye gaze, eye tracking, we will look at facial expressions, etc. So we are going to build a digital biomarker suite of data collection for every subject. You walk into a clinic or a center, within 20-25 minutes we should do everything so that we can do some simultaneous stuff. So we are designing that protocol. What we want is minimum viable protocol, MVP. MVP has many things, minimum viable product, minimum viable whatever. So we are saying minimum viable protocol which we can scale. We want 100,000 people to take this, and then that data, if I have blood samples of that, I do proteomics, I do genomics, I do all the other analysis, it's a huge data set for you to mine the data, okay? It's a multimodal curve fitting problem, if you may call it, correct? So this is the goal. So we are trying to build that. So if you look at what CBR wants to do, we want to do prediction, right? So we want to build robust risk scores and diagnostics. So there is a difference between a biomarker for diagnosis and a risk score to predict the possibility of you losing your cognition abilities. And of course you need to look at dynamic. Then intervention, fine. Now that we say that you have some probability that you will you are losing your cognition, then we need interventions. So lifestyle based, pharmacological, apps and devices, because every faculty of cognition is a composite outcome, correct, of a task. So what a professor's cognitive abilities are different from a farmer's cognitive abilities, because what they require for their daily life is what it is. So we should have high frequency sampling of all these parameters, so that, and that can be possible only through wearables and digital technologies what we have. So it's a very interesting possibility of that. And then what happens? All that can happen with data. So data has to be, now we are creating a platform which is a collaborative platform where people can collaborate. Data cannot leave the owner because of health data. And then we run this data analytics and multi-omics approaches. And this is, where will the data come from? From the cohorts. So this is our CBR's goal, and this is what we want to do the way forward. We have done some work, preliminary findings. I'll only flash, I know I'm off time by a minute. So people have found that looking at the MRI images, they have lesser volume of the hippocampus for people with MCI. Okay? So this has been observed in our cohort, whatever preliminary data we have. Okay? The other one is social connectedness. So this is CNI, so I thought social network is important. Okay? So there is a measure called Cohen's Social Networking Index, and this was, so let's say assume that you have two people, you meet, one person meets the same number of people, okay? Both of them meet the same number of people regularly. The one which is meeting a diverse set of people has better cognitive abilities compared to the one who is meeting only similar kinds of people. So you need to start socializing with non-engineers, meet artists, lawyers, whatever, okay? Not right now, but maybe later. This is not WhatsApp social networking, this is personal working, okay? So it is important, social networking. Hearing loss. Hearing loss is the first marker for dementia. The reason is, if you are not hearing, you are not part of conversations, and your social isolation begins. Okay? So if somebody elderly is in your family and you feel that they are not using hearing aids, please encourage them to use the hearing aids because their cognitive functions are there. So this is what it is. And then there are some protective factors. So people who knew multiple languages, most of Indians know multiple languages. They have what is called cognitive reserve. They have better resilience towards decline. Okay, and this was a very small cohort of 30 musicians we have in our cohort, and they're trained in Carnatic music, and for at least 5 years they had better cognition because they were musicians, etc. They also had higher volumes of gray matter in various brain regions. So white matter is the myelin sheath part. Gray matters are all the synapses. Okay? Just think of it like that. And regular physical activity is very important. So there are 3 things which you should do for healthy aging: diet, exercise, sleep. Because only when you sleep, the glial cells are in action to clean away all the debris. You should have good quality sleep. I know all of you are all-nighters and all those things. It may be okay now, but not good habit. Sleep well. It doesn't tell you that you have to sleep in the night. You have to sleep well for some time for the cleaning to happen. Okay. Anyway, this is about CBR. We were formed in 2014, registered as a not-for-profit society, and Pratiksha Trust funds us. And this we have governing board, inter and advisory board, etc. That's it. Thank you.
Thank you very much, Professor, for accepting our invitation and delivering such an interesting talk. Many of your suggestions will be useful for us. So, any more questions?
Yeah. So this must have been an unusual talk where no equations were there, right?
You mentioned about high frequency sampling and low frequency sampling. How is low frequency sampling was sort of clear from the introduction. How is high frequency sampling useful for this low frequency interpretation?
Correct. Very, very good question. Ultimately, the goal is for us to measure the parameter called mild cognitive impairment. So what is happening is, what we have observed, this year they are normal in the cognitive test. The next year they come, they already are impaired. But that has not happened suddenly. It is happened gradually during the whole year. So we want to understand when they turn that MCI, right? We don't know. It's we don't know when that has happened. But if you start measuring these things at a higher frequency, it could be daily, it could be weekly, whatever, we have to see. If there are variables, we collect data as it comes. Then we will be in a position to have that higher number of samples to estimate. Yeah. 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 The reason why we want to do high frequency also is the ability to scale. See, I only have few hundred people with MCI, okay, out of 10,000 or 12,000 people. Now, that means I have more healthy people. So how is my classifier? What is my model? My ground truth, I have only few hundred. I need thousands of ground truth values of MCI to even do some variable is important for us to scale
Scale it, yeah.
and get that information and then fit that to my MCI estimation.
So variable
Plus
measurement
cognitive test.
plus some kind of a decision. If there is a crude
Yes.
decision making, then it will reduce your data, useful data.
Correct. And we need to know which of these sensory data is really required. We have all kinds of things. As an academic researcher, we want to see a signal everywhere, right? We want to do MRI, very difficult to get people to get into MRI. But without MRI, can I do something with other markers where I can do, right? It's a proxy maybe, but we need for screening.
It's an attempt. It's an attempt.
So it's a data-driven approach to understand the dynamics of or the disease dynamics and at a population level. Okay, that is important. Scale, population level, epidemiological studies is important for us to understand this. So it's a social setting. At this for Alzheimer, for dementia, we are the ones who are running these large cohorts. There are other cohorts, small cohorts. NIMHANS has disease cohorts. NBRC does something, yeah, but they don't have so much of cohorts. Where? In CBR. These are pictures in CBR. Yeah. No, they are available not in one place. Yeah. Each one is a off-the-shelf equipment. Now we are trying to get a different version of digital biomarker suite. Speech is something which we really have to do, because the people have shown that speech variations in speech can be detected for neurodegeneration. So we want to collect speech. That's why Prashanta is going to help us. Not Vani, he has done work with NIMHANS to classify neurodegenerative diseases, ALS and non-ALS, etc., with Dr. Nalini. So for the last many years, he has been collecting data from patients. So he has a protocol. Maybe. I Casper. Okay, yeah.
Sir. Yeah, so, I mean, there has been this news regarding gravity aging hypothesis that came to quite some buzz because of Dipendra Goyal. He was wearing this temple device that continuously measures the blood flow in the brain and all. So any comment on that? I mean, many people are saying that it's a hoax, like there's nothing, no scientific evidence to it. But
I don't know. That is just one sample. What you have to understand is, some cells can divide still in the body multiple times, more than, the range is 40 to 60, correct? So some of them can go beyond, stem cells, etc. Stem cells are the ones which allow division. That's how our body from a single zygote goes into this, right? So the telomerase is important because it doesn't shorten the telomere. Okay, cancer is the one such thing. It is able to divide in a very rapid manner because it is in the presence of this telomerase enzyme, which doesn't, which stops the shortening of the telomere. Okay, so people are thinking about that kind of an activity where can I see whether I can prevent shortening of the telomere. Okay, then we have more healthy cells, and they can go do repair. Skin cells they have more division number, so they can do, because they go and repair things.
and is CVR also targeting towards this active monitoring telemetry kind of observability stuff, like where you have variables or the centers?
Yeah, right now we are also thinking of home-based stuff, so that is where these things happen. Currently, we give actigraphs, wrist actigraphs, to 20 people in this rural cohort, and for 7 days they wear it, they bring it back, we understand their sleep patterns, right? So that is what it is. But we want to see whether the smartphone can be a good device to do many things, and then that will upload data into the system.
Like I had a question regarding this brain only. Like, can we do some brain imaging, brain mapping by using some integration of fMRI and CT scan? Can you elaborate on this topic of integration of fMRI and CT scan for brain?
Correct? You can't move in MRI. So these are difficult things. So now there is something called low field MRI machines are coming up, 0.5 Tesla. What we have is 3 Tesla. So low signal to noise ratio because the magnetic field is low. So very nice signal processing problems to see whether you can extract data from that part, right? So yes, the answer is, the goal is not just to get one kind of domain and estimate, you will get one estimator. But if I add more domains, how well will be my estimation of the parameter. So if I look at some parameters using MRI, what can I do? If I look at OCT, what can I do? Like that. And then ultimately you have to fuse things.
Means more set of data will give
Yeah.
more accuracy.
Exactly. Ah. Good question. I was
Or is
waiting
it
for
like
that.
made open?
Not
for-
yet. We will make it open after a certain period of time, where CBR people will fight for getting the first results out, and then we will open it.
And the modeling aspects, the results that you showed in the last 4 to 5 slides, I hope that they're all from the cohort
Yep.
collected, right?
Yeah.
What kind of models were used? Is it like the statistical analysis?
Biostatistical models.
Biostatistical. Okay. Thank you.
Not complicated signal processing models yet, but there is a lot of scope and richness in the data which can be exploited.
And the participants who volunteered to participate in the cohort, were they randomly, like, were
Yeah.
they sampled based on some criteria, or was it like 45 years, anybody interested to
45
participate?
years, they should have been in Bangalore at least for 5 years.
I see.
Because we don't want migration happening, right? If I want to follow them up, I'm assuming that they will be here for in the future also, right? But many of them have left. They've gone to their children's in US, etc. Some of them have passed away during COVID. So we have almost 400 people who passed away in the rural cohort, and maybe a few tens in the urban cohort. And some of them have left the city. So then we lose. So a lot of missing data problem we have. Also, not all the battery is done for everybody. Somebody says, no, I want to do MRI. I won't do MRI because I feel claustrophobic. Our MRI sequence is 45 minutes. They say, no, after 10 minutes I will walk off. So I will only have the first 3-4 sequences I have. That is all I will have MRI. So it's a multi-dimensional. If I put all of them, the vectors are not the same length. They are different lengths. So, and missing data, some of them will not come. So
No, the only thing is because CBR is located in IASC, and I hope many professors would have volunteered.
yeah.
Isn't a cohort biased towards
Yes, good question.
high
Initially,
educated?
Yeah. So initially it was all professors, but later on we have gone to flats, gated communities, and recruited people. People come from all over. We provide transport for them to come to CBR. So we pick them up, we drop them, we give lunch, we do all that.
Okay.
So they come in the morning, we take fasting blood, we give them breakfast, and then like that. So
Thank you. Great
and a
work.
token amount of 200 rupees we give. We can't give more because ethics committee doesn't allow us to pay more, because that should not be the incentive. The incentive is for them to contribute to research and advancing science.
Yes, one question was there, like, is there any direction towards bio footprint on brain? Like, it's already known that it is very difficult to do brain surgery, especially for people having some issues related to mental health and all. So is there any bio footprint research towards that, like biomarkers, like by using some optical source we can make some kind of spot so that becomes a reference point, and then based on that reference point the different regions are identified, and then that particular bio footprint can automatically get rid of after some days or something like
Okay,
that.
you are trying to put a reference sensor or point, etc. Right
yes,
now what happens is, in an MRI, if you do MRI, there is a map, there is a reference pattern of the MRI, because the volumes of our brains are different from Caucasians, the sizes of our skulls are different, because it's based on the genetic part, right? That is what is done. We don't need to implant anything. For Parkinson's disease, there is something called deep brain stimulation, where an electrode is put in the region of the brain, and it is there throughout your life. It can last, the battery can last for about 20 years, it costs about 20 lakhs, and
I mean to say biospot, in which we
biospotting. No,
biospot, like for
I
doing
don't
some
know
operation.
much. We are not doing, and I don't think it is really required right now. I think they have other tools which are reasonably good for surgery.
I hope Professor has addressed all your queries. So, online questions. Okay, seems like there
What
are
should
some
I? Yeah,
online
sure.
questions.
You have to ask.
So, yeah, we have an online question from Gargi Mahapatra. She says, thank you, Dr. Hari, for the talk. For all of the cohorts you discussed, both in urban and rural population, do you have any sex-specific analysis? As it is known that females are diagnosed with MCI and AD at a significantly higher rate than males. Is there any data on that for your cohorts? What about extremely high pollution levels like we are experiencing in Delhi area?
Okay, yeah, good question. We, in all our both cohorts, our gender, there is a gender balance. So we do have equal number, almost equal number of male and female. Elsewhere, it has been found that women are more prone to dementia than men due to hormonal issues. Okay, we have found that also here. Even though cohorts are small, but we do have, but that is for dementia. We are now looking at MCI, correct? We don't have disease people. Disease people, you have to go to the hospital, but we are seeing a trend of women being subjected to. The other thing is, in rural areas, there is a lot of exposure to firewood in the kitchen. They still use, they don't use CNG or whatever. So they use firewood, there is a lot of pollution. So there is a particulate matter of 2.5 breaches the blood-brain barrier, and it can cause trouble for that. So therefore, people exposed to pollution, and many of them are women who are in the exposed to that pollution. So there is another factor there.
Thank you once again, Professor. So, we have arranged refreshments at Coffee Head. Please continue your conversations there.
Thank you very
all
much.
and have a great evening.
Thank you.